Professor Amit Dutt. Amit is a professor at Department of Genetics, University of Delhi South Campus, and is also a principal investigator at Advanced Center for Treatment, Research and Education in Cancer at TREC Tata Memorial Center, Mumbai. He is spreading a pioneer research team which is revolutionizing the cancer treatment through the lenses of genomic intricacies. His topic of today's presentation is genetic alteration and microbial dysbiosis that underlines lymph nodal metastasis in tongue cancer. Professor Amit.
Thank you. Very good afternoon, everyone. I start with, that's correct, that's my slide. Many thanks to the organizers, especially Professor Gopinath, for having me over here. As what Rupesh has already just kind of like mentioned, I'm now a faculty at University of Delhi South Campus, Department of Genetics. But however, the work that I'm going to be talking about today would be primarily from the work that I have done till last month at ACTRAC, Tata Memorial Centre, Mumbai. So I cannot see the slides over here. May I? Excuse me. यहाँ पे slides नहीं दिख रहे. Oh, okay. So today I'm going to talk about primarily oral cancer. I will be restricting myself to, and I'm going to talk about the genetic alterations that underlies it and how basic biologists, the findings from the basic biologist laboratory, how it can get translated and how it can impact not just the advancing the understanding of the underlying biology, but how it can get translated onto the clinics and benefit the patients as well. And this is the work that has been done in collaboration with a clinical surgeon friend of mine, Dr. Sudhir Nair. So before I really start, I wanted to set up a premise. I'm not introducing this crowd to the oral cancer, which happens to be the tobacco-associated cancer to be about like 40% of all the malignancy that what you tend to get to see here in India. It's one of the most predominant cancer, but the statistics is something that I'm not going to be talking about today due to the brevity of the time that has been given, 15 minutes. So, introducing the problem, essentially in oral cancer, primarily one of the subtypes that my laboratory is interested in, that is in the tongue cancer. There are multiple subsites. Our laboratory is primarily been restricting ourselves to the tongue. Tongue is something that affects the younger generation as compared to the buccal gingiva or other different sorts of oral cancer. So if it is being detected at an advanced stage, you would see that the survival is significantly low. It is 20% and not much can be offered over there. However, as what they say in the Tata Memorial Centre, law says that cancer can be cured if detected early. So what happens when an oral cancer is one of the textbook example of cancers which can in principle be detected early given the accessibility to it. So however, if you would see in early stage tongue cancer and oral cancer, what essentially lies that there are two different types of it. One is that if there is a metastasis and something which is called as an occult metastasis, which happens in 30% of those, and that is not very apparent to the clinicians, to the surgeons primarily. And then there is 70% of the patients who do not have an underlying occult or cryptic metastasis. And what does it kind of like lead to? Those folks who are having an underlying cryptic metastasis, the survival goes down to 40%. And the 70% of patients, as what you can see on the left side, are those guys who do not have the cryptic metastasis, and there the survival by itself is about like 80%, the 5-year survival. But the question is, how do you make a distinction between them? That who are the folks who are falling in the 70% group and who are falling into, because the current regimen, I mean, all the imaging facilities which are available, even the sophisticated ones, they are not able to make a distinction, or they are not able to identify those cryptic metastases which is happening in the oral cancer. So, in fact, there was a clinical trial that was done by the director or former director of Tata Memorial Centre, Tata Memorial Hospital, Dr. Anil T. Cruz, where he kind of like showed that indeed that if you are doing a therapeutic surgery, that is removal of the tumor along with the lymph nodes, which has huge morbidity associated to it, has a better survival. So now what is happening is that all the patients, even the 70% of the patients who actually did not require surgery, did not require the lymph nodes to be removed, but there is no way to identify them, so they were going to that huge morbidity which is associated with that therapeutic. surgery rather than elective surgery rather than doing a therapeutic one. So in elective, they could remove the primary tumors, and along with that, all the lymph nodes which are associated with it. So there comes the role of a biologist. What would a biologist do over here? The biologist would want to would want to kind of understand and to look for some molecular markers that would help or that would aid a surgeon to make a decision which one to go for a surgery and which one perhaps not to. That's what was the initial problem that we kind of like got started with. And my laboratory is a cancer genomics lab in which we have a strong genomics arm, computational arm, and along with that we also have a strong functional group. So this is a textbook picture that is from an NGS platform. And essentially what it is kind of like showing is that it's so disruptive to an extent that now all the different kind of alterations that what you know that underlies the cancer genome, whether it is mutation, whether it is copy number alteration, whether it is translocation, or unfortunately I do not have a pointer to show you, or whether it is sequences which are not human in origin, such as those which are associated with pathogens perhaps, those all which in conventional way required different technologies. For mutation you would ideally be doing a Sanger sequencing. For copy number alterations you would ideally be doing microarrays. For translocation events you would be looking for FISH kind of, you need to have an a priori information though. And similarly for pathogens, if you're really wanting to look for, you would be going for a directed PCR event. But that's where the disruptive nature of NGS comes in, that in one single go, in one typical run, you would be able to identify all of these kind of like alterations. So what I'm going to do today, I'm going to talk about that how this genomic approach would lead or would allow a biologist to identify these alterations of different kinds in oral cancer and how that can inform or lead to more of a clinical translation. So this is one of the first study that what we were kind of like doing, that was with 151 odd samples, but we went on to do like much more but much more higher number. And essentially what it is, this is so these are all, if you would see that they are equal number of, a comparable number of node positive and node negative. These were pathological node positive and node negative samples where we knew that those guys are node positive and these are node negative. And essentially what we were trying to do, we were trying to understand if there were any genomic biomarkers that will allow us to make a distinction between them. And then what did we do? So that's to begin with. For the first thing that we were doing is that go ahead and perform an whole exome sequencing. So we went ahead and performed an whole exome sequencing, and what did we find? I'm going to give you an overview rather than getting into the details of it, because I see that it's late in the afternoon as well. So essentially from the whole exome sequencing we found that what is shown in the red block over there are the hallmark alterations of head and neck cancer. So if you are doing any kind of these sequencing, you would always want to benchmark it against something that, okay, these alterations that you were expecting to find is something that what you're finding in the Indian oral cancer tumor or not. That would kind of make you more confident about your own data and about the algorithms and everything that what you're following up. So as what you see, there were no surprises in here. However, the surprise lies in the eyes of a beholder, I would rather say, of the intellect of the student, the critique nature of the student. So the student, Padma Shri, what he did is that he finds that there were alterations in notch 1. Those times, notch 1 was known to be a tumor suppressor gene in oral cancer. What Pavan found, the mutation, the nature of those mutations in oral cancer were not inactivating alterations. Those were not deletions either as well. What he found that they were missense alterations. And when and also he found at the copy number space that there was slight more of an amplification. There was a bit of a baffling, and that was something counterintuitive, that the tumor suppressor gene is amplified and the tumor suppressor gene is not having inactivating alteration. So that comes the functional arm. Pavan takes these alterations which were found, which were derived from the patients, and then he takes it onto the bench and then performs, overexpresses those cells in cell lines which were established for Indian origin patients, and he does some elegant biochemical and cell-based assays, and and eventually, cutting a long story short, what he found was that Notch1 alterations are also activating in oral cancer. And it went on when we performed the clinical correlation, it turned out that those guys who are tobacco chewers tend to have, and they are young, they tend to have more of these notch alterations. And why it was an important and a big news is because notch alterations pathway inhibitors are already there in the clinical trial and approved for other different diseases. So essentially it would be adaptation of those notch inhibitors into the oral cancer was one of the major thing that was coming up. But we were primarily interested in understanding, are the clinicians able to make a distinction between what is lymph node positive and what is lymph node negative? So here it was associated with lymph node positivity, associated with tobacco chewers. Another student joins in at that point of time, Bhaskar Dharawat, a very talented one. Bhaskar Dharawat was now looking at a similar kind of like data set in the lab generated at a much larger cohort, and what he was looking at is the copy number alterations. So Pawan was looking at mutations from an NGS data set. Bhaskar is looking at copy number alterations, and what did he find? What he found is something far more interesting as well. He found that there is a matrix metalloprotease. And just to kind of like mention about the Notch story, by the time we were completing that story, there were already two Nature Genetics back to back talking about the Notch to be an oncogenic role in oral cancer. So now it is well established that the Notch alterations, depending on the context, could be tumor suppressor as well as an oncogene, even in oral cancer, the way as what it has been shown in the breast cancer as well. So we were late in there. So, but Pawan Upadhyay, he found that matrix metalloprotease to be significantly amplified, significantly associated with nodal metastasis. And not only that, he went on to kind of like look at the transcriptome analysis, and what he found is that they were also overexpressed. There was a strong correlation between amplification and overexpression, and that was strongly associated with the nodal metastasis. The lab has a strength in functional biology. So what does Bhaskar Dharawat do? Well, Bhaskar Dharawat takes up onto the cell, does huge amount of knockdown, knock-in experiments, and he goes on to kind of show. I don't think I will be able to show you because this doesn't work, and there's a movie over here. So Bhaskar went ahead and established an orthotopic mouse model along with our postdoc, Dr. Ashwin Butle. I won't be able to show you because I don't think it will go to the next slide. Yes. So essentially this was the procedure. It was a small snippet that showed you that how cells are being injected to a few millimeter of tongue into the mice, and how an orthotopic mouse models are being made a tumor straight onto the tongue. So this is where on the lower panel you can see that where the cells after injection, and then exactly after 26 days what we tend to get to see is that there is a bulge, there is a tumor that kind of like forms in these orthotopic mouse models. Lab is very much into developing these kind of like mouse models, not just into the oral cancer or tongue mouse model. We have developed orthotopic lung cancer mouse model by directly injecting the cells straight into the intercostal muscles and the tumors forming into the lungs, not by the skin xenograft assays. Similarly, the lab has been developed now a very sophisticated cervical cancer orthotopic mouse model directly onto the cervix. I don't know if these kind of like mouse models exist anywhere else like in the country. Similarly, in the breast we have developed these orthotopic mouse models. However, my today's talk is primarily for, my time is up, but I'll take me a few more minutes perhaps. Thank you. So I was back onto the oral cancer mouse model. That is the tongue cancer mouse model. So by doing all of these, what essentially Bhaskar Dharawat showed, he dissected the whole pathway of that how MMP10, when it is amplified, what is upstream to it, how it is being regulated, not the focus of the talk today, but then he went on to kind of like show that there was a significant metastasis that was associated when the MMP10 was overexpressed. Another very talented graduate student, Sanket Desai. Sanket Desai is a computational biologist, a hardcore computational biologist. So he joined the lab and he started looking at the computational subtraction of these kind of data sets, which comes out from the NGS platform, and he throws out everything which is human, and which is where all the other students were interested in, and he was looking for something which is like non-human traces in the human genome. And what did he find? He found something very fantastic, landed up into a very interesting finding. He found that there was most of the oral cancer, the tongue tumors primarily, they are devoid of HPV in the Indian subset. They are primarily driven by tobacco. 20% of Caucasian population, the TCG data set, is driven by the HPV. Here it is primarily the tobacco. What did he find is that there is another pathogen, that is a fusobacterium. An oncobacterium, which is known to be associated in colorectal cancer. He found a significant prevalence in the Indian tongue cancer mouse and Indian tongue cancer patients. We are studying in Indian human cancers, not mouse cancers. So, and then when he looks into the TCGA, he finds that the fusobacterium are also present in the TCGA, and there is a mutual exclusivity. Here is a set of patients which are HPV positive, they do not have fusobacterium. Here is a set of patients who are having fusobacterium, totally mutually exclusive, they are not positive for the HPV. There are very few samples which were doubly positive. And then starts the whole thing that we are interested in fusobacterium, going ahead and doing the co-culture experiments and trying to show that these fusobacteriums are indeed associated with, there was an extracellular lymph spread which was associated with fusobacterium. So what did we talk about? We talked about the mutations, the Notchman mutations, we talked about the MMP10 alterations, all associated with nodal metastasis, we talked about the fusobacterium, and the game changer is the fusion event. Bhaskar, this was just recently published in NBJ Precision Oncology, where Bhaskar, I'll cut the story short, I will just go straight into, we were also looking for translocation events. So as you know, that translocation events such as ALK, ROS, NTRK, those are game changers out there in the precision medicine. In fact, the NTRK alterations have been shown to be like pan across different, it is tissue agnostic, it's approved, the inhibitors for the NTRK. So wherever these fusion events are the rare of the rarest kind, it's not very easy where a chromosome A breaks at a specific point, where a chromosome B breaks at a specific point, A and B in frame comes and like fuses. What Bhaskar Dharawat, along with Dr. Sanket Desai, what did he find is that there is a novel translocation event between chromosome 11 and chromosome 5 in oral cancer, and that was not just by the transcriptome sequencing. There are a lot of artifacts when you do the transcriptome sequencing. They were able to then, they found that in 4% of the patients, essentially it's 5.6% now we know of, in the Indian patients where we have this fusion transcript. The good thing is the gold standard in the fusion transcript and in the translocation event is that you have to show it onto the chromosome. You have to show to the resolution of a single base that if there is actually the breakpoint that you will be able to identify, and that's what they did. They went ahead performing whole genome sequencing, a huge and a tremendous and a very tedious and very dogged effort required to map the exact translocation site onto the chromosomes. And then of course validation, and when we looked at the TCGA, whole genome sequencing and the whole transcriptome sequencing, we found that there were about 1.2% incidence, which is where most of the fusion transcripts, as what you know, and lies. I mean, an NTRK occurs at 0.2 or 0.1% frequency. So the game changer is that these fusion events, novel fusion event, which has not been characterized, just 30 seconds more, sorry for doing this, these were significantly activating. I mean, we know that fusion events, rare of the rarest kind, they are very activating in nature, they are very oncogenic, and these were indeed, there were multiple points. For a complex fusion, we have deciphered the whole loci of how these reciprocal fusion events happen. It is already published in NBJ Precision Oncology very recently, a week back actually. We went on to kind of like show that indeed that these were activating these fusion events. Earlier in the talk we heard about the beta-catenin and we talked about the Wnt signaling. We found that this fusion event is actually targeting the Wnt signaling. What it is doing is that when the fusions are present, you can see that there is a localization of the beta-catenin in the nucleus. I'm sorry, I'm not able to take you into the details. These are two different oral cancer cell lines established from Indian oral cancer patients as a legacy from the Tata Memorial Center. We could show in both of them that when this fusion transcript is overexpressed, it leads to its transformation. Then a series of assays, whether it is proliferation, whether it is other different activating aspects of cancer, those were found to be associated with these fusion transcripts. Again, this is the first fusion transcript that has been characterized to this extent in oral cancer, which is, but the most important thing is that they were forming tumors in the mice as well. So that's the gold standard. The most important thing is that the student comes along that there is an inhibitor out there, and there is an inhibitor out there, the Wnt signaling inhibitor, which is essentially a dewormer, something for deworming that is already FDA approved and that targets Wnt signaling. So again, this is the Bhaskar Tharawat's gift, where he digged out all the literature. I knew nothing about it, and then what he found that there is an FDA approved drug. Let's go ahead and we have the mouse models. Let's go ahead and test it.
You will have to wind up, please.
Yes. So I'll cut short this. So we did wind up. I'm winding up. So we found that there is a pyrivium pamboate Wnt signaling inhibitor. That is something that significantly inhibits this. So if there is a patient who is positive for this, is likely the clinical trial for these effects are currently being undertaken at the Tata Memorial Center. So now, cutting a long story short, is that the algorithm is being developed where a clinician would be able to figure it out with the MMP10, with the Notch1, with the Fusobacterium, and with the translocation event that which one to go for surgery or not. These are the students from the lab, primarily all of them, and Akshita Chaudhary today over here sitting in the group as my lab is located. Thanks to Tata Memorial Center, the DBT Wellcome Trust, India Alliance, DBT VNCI, CERT, BST, Terry Fox Foundation, for all the generous funding that I've received. Thank you very much. Sorry for explaining.
Thank you, Dr. Amit.